Adaptive immunity. Adaptive immunity is the body's targeted response to infection. It is activated only after the first lines of defense, such as physical barriers and inflammation, have been breached. Unlike the immediate and general inflammatory response, adaptive immunity is specific, inducible, long-lived, and possesses memory. This means it not only attacks particular pathogens, but also remembers them, preparing the immune system to respond more rapidly and effectively in the future. Two key elements form the foundation of adaptive immunity: antigens and lymphocytes. Antigens are molecules found on the surface of microbes, infected cells, or abnormal tissues. These molecules are the precise targets of antibodies. The lymphocytes responsible for adaptive immunity include T cells, which mature in the thymus, and B cells, which develop in the bone marrow. Adaptive immunity operates through two major arms: humoral and cellular immunity. In humoral immunity, antibodies circulate in the blood and bind to antigens found on bacteria or viruses. Cellular immunity is mediated by T cells, which patrol the blood and tissues to defend against infected cells and abnormal or cancerous cells within the body. There are two types of adaptive immunity: active and passive. Active immunity develops after exposure to an antigen, either through infection or vaccination, and it produces long-lasting protection. Passive immunity, in contrast, involves the transfer of pre-formed antibodies or T cells from another source, such as from mother to baby across the placenta. Passive immunity provides immediate but short-term defense. Antigens are molecules that bind to antibodies or to receptors on T and B cells. But not all antigens cause an immune response. Immunogens are a special category of antigens that not only bind to immune cells, but also stimulate an immune reaction. All immunogens are antigens, but not all antigens are immunogens. For example, small molecules called haptens only become immunogenic after attaching to a larger carrier molecule. Clonal diversity refers to the generation of a wide variety of B and T cells that can recognize almost any foreign antigen. This process takes place mainly during fetal development in the body's central lymphoid organs. The result is a large population of immunocompetent cells, cells that are fully capable of responding to an antigen when the time comes. B cells develop in the bone marrow. During this time, they undergo production, proliferation, and differentiation. Each B cell forms a unique receptor that recognizes a specific antigen. If a B cell reacts against the body's own tissues, it is eliminated in the bone marrow through a process called central tolerance. This is essential to prevent autoimmunity. T cells develop in the thymus, which is the primary organ for T cell maturation. T cells form receptors that recognize specific antigens and then migrate to secondary lymphoid tissues like the spleen or lymph nodes. Like B cells, T cells also undergo central tolerance to ensure that they do not react against the body's own tissues. Both B cells and T cells use specialized antigen receptors to identify and respond to foreign invaders. These receptors are uniquely generated during the early development of the cells and remain highly specific. The B cell receptor allows the B cell to recognize antigens in their native form, while the T cell receptor requires the antigen to be processed and presented by another cell. These receptors are what enable adaptive immunity to be precise and personalized. Clonal selection is the second phase of the adaptive immune response. It begins when antigens are processed and presented to lymphocytes by antigen-presenting cells. These include cells like dendritic cells, macrophages, and B cells. Once an antigen is displayed on the surface of an antigen-presenting cell, it initiates a complex network of cellular interactions that lead to immune activation. The clonal selection process is triggered when T cells and B cells come into contact with their specific antigen. The antigen must be presented on the surface of an antigen-presenting cell, or APC. Abnormal cells also display antigens through molecules called major histocompatibility complexes, or MHCs. This interaction causes B cells to differentiate into plasma cells that produce antibodies, and T cells to become effector cells, such as cytotoxic T cells. The major histocompatibility complex, or MHC, is a group of glycoproteins found on the surface of nearly all human cells, except red blood cells. These molecules are also called human leukocyte antigens, or HLAs. MHC class I molecules present antigens from inside the cell and are recognized by cytotoxic T cells. MHC class II molecules present antigens from outside the cell and are recognized by helper T cells. This distinction helps the immune system target the right type of threat. Through clonal selection, the immune system custom builds a response to each specific invader. It ensures that only the cells capable of recognizing the presented antigen are activated. These activated lymphocytes then multiply and carry out their protective functions with precision. The effective immune defense depends on collaboration between different cell types. These interactions result in the creation of two main products: effector cells that fight infection and memory cells that provide lasting immunity. This process depends on three types of signaling: antigen-specific recognition by B cell or T cell receptors, communication between cells, and the release of cytokines, which help coordinate the response. T helper cells, or Th cells, play a central role in adaptive immunity. When an antigen-presenting cell presents an antigen, the T helper cell binds to it and becomes activated. Different subsets of helper T cells serve different roles. Th1 cells support cell-mediated immunity. Th2 cells help stimulate antibody production. Th17 cells activate macrophages. And regulatory T cells, or Tregs, suppress immune responses to maintain balance. Superantigens are a dangerous type of antigen. Unlike normal antigens, they bind outside the usual recognition sites on both the T cell receptor and the MHC class II molecule. This bypasses normal immune regulation and activates a massive number of T cells at once. The result is an overwhelming release of cytokines, leading to a systemic inflammatory reaction that can cause fever, low blood pressure, and even shock. Cytotoxic T cells, also known as TC cells, are essential for killing infected or cancerous cells. When a TC cell recognizes its target antigen presented by an infected or abnormal cell, it becomes activated. These effector TC cells then directly kill the affected cell, using specialized enzymes to trigger cell death and stop the spread of infection or malignancy. Once activated, cytotoxic T cells become highly specialized killers. They patrol the body, seeking out infected or cancerous cells that present the specific antigen they were trained to recognize. When they find their target, they release molecules that trigger programmed cell death, or apoptosis. This helps the immune system eliminate threats without causing excessive damage to surrounding tissue. When a B cell encounters an antigen for the first time, only those cells with receptors matching that specific antigen become activated. These B cells undergo rapid proliferation and differentiation. The result is the formation of plasma cells, specialized cells whose main job is to produce and secrete large quantities of antibodies tailored to the detected antigen. In addition to producing plasma cells, clonal selection also creates memory B cells. These long-lived cells remain in the body after the initial infection has passed. If the same antigen appears again, memory B cells respond rapidly, producing antibodies with little delay. This swift secondary response is the cornerstone of lasting immunity and why vaccinations are so effective. Both B cells and T cells produce memory cells during an immune response. These memory cells are inactive until they encounter the same antigen again. Unlike naive cells, memory cells don't require further training or differentiation. They are primed for action and can quickly convert into plasma cells or effector T cells, ensuring a rapid and efficient defense on repeat exposure. Antibodies, also called immunoglobulins, are proteins produced by plasma cells. There are 5 major classes: IG, IgA, IgM, Ig, and Ig. Each class differs in structure and function. These antibodies circulate in the bloodstream or secretions and are responsible for recognizing and binding to antigens, neutralizing threats, and triggering additional immune actions. Let's begin with immunoglobulin G, or Ig. It is the most abundant antibody in the body, making up around 80 to 85% of all circulating antibodies. It plays a major role in protecting against infection and is unique in its ability to cross the placenta, providing passive immunity to the developing fetus. Immunoglobulin G, or Ig, is the most abundant antibody in the human body, making up approximately 80 to 85% of all circulating immunoglobulins. It plays a critical role in protecting against infections by neutralizing toxins and viruses, opsonizing pathogens for phagocytosis, and activating other components of the immune system. A unique feature of Ig is its ability to cross the placenta, providing passive immunity to the developing fetus and newborn. This transfer of maternal antibodies helps protect the infant in the early months of life before their own immune system is fully developed. IgA is found in 2 forms. One circulates in the bloodstream, but the more important version, secretory IgA, is present in mucosal secretions such as tears, saliva, breast milk, and mucus. This antibody plays a key role in protecting the body's surfaces from pathogens and is specially structured to resist degradation by enzymes, helping it function effectively in secretions. IgM is the largest of all antibody types. It is structured as a pentamer, meaning it has 5 antibody units joined together. IgM is the first antibody produced during the initial immune response and is the earliest antibody synthesized in newborns. Its large size allows it to bind strongly to pathogens and quickly activate the complement system. Ig is found in very low concentrations in the blood, and its role is still not completely understood. What is known is that Ig acts as a receptor on the surface of B cells and helps initiate B cell activation when antigens are first encountered. Ig has a very specific role in immune defense. It is primarily involved in protecting the body against parasitic infections. However, when it mistakenly reacts to harmless environmental substances, like pollen or pet dander, it triggers allergic reactions. Ig binds to receptors on mast cells and basophils, promoting the release of histamine and other inflammatory chemicals. When Ig antibodies bind to mast cells, they prepare the body to respond quickly upon future exposures. If the same allergen enters the body again, the mast cells release large amounts of histamine, resulting in symptoms like sneezing, swelling, itching, or even anaphylaxis. This mechanism, while protective in some contexts, is also at the root of allergic disorders. During an immune response, B cells begin by producing the IgM and Ig classes of antibodies. However, as the response matures, these B cells can undergo a process called class switching. This allows them to change the type of antibody they produce from IgM to Ig, IgA, or Ig, without altering the antigen specificity. In other words, the B cell continues to recognize the same pathogen but gains the flexibility to tailor the immune response based on the location or type of threat. Class switching creates a diverse population of plasma cells that can produce a range of antibody types to better defend the body. Antibodies are Y-shaped molecules made up of 4 polypeptide chains, 2 identical light chains, and 2 identical heavy chains. The arms of the Y, called the antigen binding fragments, or Fab regions, are the parts of the antibody that recognize and bind to antigens. These sites are highly variable and determine the antibody's specificity. The stem of the Y is called the crystalline fragment, or FC region. It is responsible for the antibody's biological activity, including interactions with other cells of the immune system. Each antibody molecule is specially shaped to fit its target antigen like a key in a lock. The antigenic determinant, or epitope, is the part of the antigen recognized by the antibody. The matching region on the antibody is called the paratope. Binding between epitope and paratope is driven by non-covalent interactions, which are strong enough to secure the match but flexible enough to allow for quick responses. This precise fit is what enables antibodies to target and neutralize specific threats. The interaction between an antigen and an antibody is based on structural compatibility. The antigen presents an epitope, its recognizable feature, and the antibody binds using its paratope. This is often described as a lock and key mechanism. Once the antibody binds to its target, it can either directly neutralize the threat or signal other immune mechanisms to take action. These interactions are crucial for orchestrating both the immediate and long-term immune defense. Antibodies protect the body in 2 main ways: directly and indirectly. Direct actions include neutralization, where antibodies block the harmful effects of toxins or viruses, agglutination, where they clump pathogens together, and precipitation, where they cause soluble antigens to become insoluble and easier to clear. Indirectly, antibodies activate other immune components. They enhance inflammation, promote phagocytosis, and trigger the complement cascade, amplifying the immune response and clearing pathogens efficiently. When antibodies act directly, they neutralize toxins or block pathogen entry into cells. When they act indirectly, they engage with other immune system players. For example, antibodies that bind to a pathogen can mark it for destruction by phagocytes or activate the complement system, which leads to cell lysis and further inflammation. Both types of action are essential for a fully functional immune defense. The secretory, or mucosal, immune system serves as the body's external line of defense. It includes lymphoid tissues that protect surfaces such as the eyes, respiratory tract, gastrointestinal tract, and genitourinary system. These areas are continuously exposed to the environment and rely on antibodies found in secretions like tears, saliva, mucus, sweat, and breast milk. The primary immunoglobulin in this system is secretory IgA, which helps prevent pathogens from entering the body through mucosal surfaces. While IgA is the dominant immunoglobulin in mucosal secretions, small amounts of Ig and IgM can also be found. Together, these antibodies create a local immune barrier that neutralizes pathogens before they can cause infection. The mucosal immune system plays a critical role in maintaining health and is especially important in early life, where passive immunity from breast milk contributes to infant protection. The body's first exposure to an antigen triggers what's known as the primary immune response. During this phase, there is a short delay, called the latent or lag phase, while clonal selection takes place and immune cells become activated. Around 5 to 7 days after exposure, IgM is the first antibody to appear. A slightly delayed Ig response follows. This first response may be slow, but it leads to the creation of memory cells that prepare the immune system for future encounters. Upon subsequent exposure to the same antigen, the secondary immune response kicks in. This response is much faster and stronger thanks to memory cells formed during the primary phase. These cells can respond immediately, producing large amounts of antibodies, especially IgG, without needing to go through full activation again. This is the basis for long-term immunity and is what makes vaccines so effective. In addition to B and T cells, other immune cells play supportive roles. Natural killer cells, or NK cells, help eliminate virus-infected and cancerous cells by detecting cells that lack normal surface markers. Complement proteins help destroy pathogens and activate inflammation. Cytotoxic T cells eliminate infected cells directly, while lymphocyte-secreting T cells release signaling molecules to enhance inflammation. Regulatory T cells help keep the immune response in check, promoting tolerance and preventing overreaction. At birth, a newborn's immune system is not fully developed. Although the fetus produces IgM on its own, levels of IgG and IgA are low. Protection in the early months of life depends largely on maternal antibodies passed through the placenta and later through breast milk. These antibodies offer temporary defense while the infant's immune system matures. Newborns also have reduced phagocytic activity and weaker complement systems, making them more vulnerable to infections. As we age, immune function gradually declines. T cell activity decreases due to a reduction in thymic size and a drop in thymic hormone production, limiting the body's ability to generate new T cells. In addition, the antibody response becomes weaker and slower. This decline in immune efficiency contributes to the increased risk of infections, cancer, and autoimmune diseases in older adults.